Cell Cycle by Jana L. Ferraz. At some point in life everyone wishes that they could clone themselves. But we can't. You know what can? Your cells. To get to this stage, the cell goes through the cell cycle.
Okay,
So the cell cycle is the life cycle of a cell. It consists of the interphase and the mitosis. The interphase comes before the mitosis and is not part of the mitosis.
Okay, so
What is the interphase? The interphase is the stage of the cell cycle when a cell is preparing itself to duplicate, so preparing itself to go through mitosis. There are three main parts of the interphase. There's G1, S, and G2. During the G1, the cell prepares itself to move into the S phase. The chromosomes in the nucleus are not yet replicated. In this stage, the cell is growing, and therefore most of the cell's lifetime is spent here. Some can stay for weeks or years.
2,000 years later.
If the cell takes too long, they enter into what is called a G0 phase. This is basically a stage of suspended animation. To get out, special internal and external signals are needed. If they get out, the cell will re-enter the G1 phase. Transition between G1 and S is called the restriction point. This is the time when the cell is committed to duplicate its DNA and engage in cell division. The S subphase of the interphase is the second part of the interphase process. It's when all the cell's genetic material is duplicated. Each chromosome is copied and results in a pair of sister chromatids. Remember that a chromosome is a strand of DNA wrapped around proteins. These will then separate when the cell finally divides. Chromosome replication happens when a DNA strand unwinds and the strands separate. Precise and accurate DNA replication is necessary to prevent genetic abnormalities which often lead to cell death or disease.
now, the S phase to the G2.
When DNA replication is completed, the cell enters the G2 phase. G2 phase is usually the shortest out of the three phases of interphase. During the G2 phase, after the G2, the cell has to divide. So now it has to prepare itself for the division process. So in G2, the changes which take place are protein synthesis, like the G1 phase. These proteins will help in spindle fiber formation, and they would help in formation of newer membrane and membranes of the organelle. So this protein is different from the protein in G1, was histone, which was required for DNA replication. Because the cell is preparing itself for the division process, the proteins that the G2 phase requires are plasma membrane or spindle fiber. The cell double checks the duplicated chromosomes for error, making any needed repairs. Mitosis is not a process that makes sperm or egg cells because that is something called meiosis. Mitosis is done to produce body cells. Answer is uncontrolled cell growth. Mitosis is a very short amount in time in respect to the whole cell cycle, but mitosis is a critical process. So let's remember PMAT, a little acronym that helps you remember. The P is for prophase, the M is for metaphase, the A is for anaphase, and T is for telophase. So remember, PMAT. In the first stage of mitosis, the chromatin condenses into discrete chromosomes. The mitotic spindle begins to form and the nucleus disappears, but the nucleus remains intact. The second stage of mitosis is where the spindle is complete and the chromosomes attached to microtubules and are all aligned at the metaphase plate. The third stage of mitosis, anaphase, is when the chromatids of each chromosome have separated, and the daughter chromosomes are moving to the poles of the cell. In telophase, the chromosomes are actually at the complete opposite ends, and new nuclei are forming on each side to make these two new cells. The nuclei are starting to surround the chromosomes on both sides. Cytokinesis is responsible for the final separation into two cells by splitting the cytoplasm.